Dear students, welcome to Tungal online classes. In the last video, we saw what is cell cycle. What are the phases that we come across in the cell cycle? As you are seeing over here, one is interphase and second one is M phase are the two major phases that we come across over here. In interphase, there we come across three phases. One is G1, which take place maximum time over here, and followed by S phase, that is synthetic phase, where the synthesis of DNA that take place, and third phase, that is G2 phase, and remaining cytoplasmic contents or the cellular contents that duplicate once again, and protein synthesis that take place in this stage. So all these are the things that we studied in detailed manner, and it is followed by the M phase or the division phase. It is called as, that is also known as mitotic phase, which contains 4 stages. So during nuclear division, right? So why? Because there we come across 2 divisions. One is called as karyokinesis or the nuclear division, and second one is called as cytokinesis. So in this stage, so what happens over here? So there are 4 stages that we come across during nuclear division. One is prophase, metaphase, anaphase, as well as telophase. Telophase is followed by cytokinesis, where the cytoplasmic division also take place equally in daughter cells, right? So these are the facts what we are so familiar with in cell cycle in the last video, and now in this video the further causes for cell division. We are going to study what are the major causes for cell division. Of course, there is a cell. Why should it divide? So there are many of the hypotheses, many of the principles explained by many of the scientists over here, and what are the major causes of cell division? Let us see one by one. Causes of cell division. So the first cause is mitogen are those substances which are stimulating the cell division itself. What are those ones? Mitogens. Mitogens are the substances which stimulate cell division itself. So what kind of mitogens they are? The mitogens like cytokinins in case of plant cell. In case of plant cell, cytokinins are one type of mitogens which stimulate the cell division. Cytokinins are nothing but they are a kind of the plant hormones or the phytohormones they are said as, right? So this is one cause. And in case of animal cell, animal cells, there are some mitogens are there. So that is epidermal growth factor. Epidermal growth factor is one factor which stimulates the cell division itself. So this is in case of animal cell. And one more is there. So PDGF is also another factor, right? PDGF is also another factor, so that also stimulate the cell division over here. So these are some of the mitogens in case of plant cell as well as animal cell. Cytokinins, EGF as well as PDGF, right? So like this, these are the mitogens which stimulate the cell division. So this is one fact. And second one is very important one of the theory, right? Cairn plasm theory it is called as. Cairn plasm theory is one of the theory which says why should a cell divide itself. Hartwig is one of the scientists. So the one who says that, so there is a disturbance in the cardioplasmic index. So due to this disturbance in the cardioplasmic index, the cell so inevitably divides itself, right? So this is the fact what he says. Hartwig is the name of the scientist, the one who says there is a disturbance in cardioplasmic index, he says. So what is this cardioplasmic index? So with the help of a formula he explains over here. So cardioplasmic index is equal to Vn divided by Vc minus Vn. This is the formula what he gives over here. So what do you mean by Vn? Vn is nothing but volume of the nucleus. Volume of the nucleus. Vc is nothing but it is volume of the cell. And so Vc minus Vn is nothing but, so it is the volume of the cytoplasm itself, right? So cytoplasm. So cardioplasmic index is disturbed over here. Due to disturbance of cardioplasmic index, what happens? Cell divides, he says. So according to himself, cardioplasmic index Ki is depending upon 1 by volume of the cytoplasm. So this is also one more fact he explains over here. Alright? According to himself, what happens over here? If the cell is very small, enough, then it contains very limited amount of cytoplasm. If there is a limited amount of cytoplasm is there, it is easily controlled by the nucleus. So whatever the metabolic activities that are taking place inside the cytoplasm, they have been controlled by the nucleus. They will be under the control of the nucleus if the cell is smaller enough. Okay, so when the cell that grows becomes larger, the cardioplasmic index is less, right? In a small cell, less cytoplasm, cardioplasmic index is high, right? So there the cell division doesn't occur. Why? Because the cytoplasmic activities are controlled by the nucleus. But whenever the cell that grows becomes larger enough, so cardioplasmic index is less, so that what happens? Nucleus cannot control the metabolic activities so that are taking place in the cytoplasm, so that it leads to cell division itself. Right? These 2 points we have to mention over here. In small cell, less cytoplasm is there, so that high cardioplasmic index is there. So that what happens? Nucleus controls all metabolic activities. Controls all the metabolic activities, so that no cell division occurs. But in large cell, what happens? More cytoplasm is accumulated over here, so that a less cardioplasmic index is there, so that nucleus cannot control over all metabolism that take place in the cytoplasm, so that division of cell occurs here. Division of the cell it leads to. So this is one theory, so that he explains, so that why a cell divides. Why a cell divides. This is one theory. And the second one is surface volume ratio. Surface volume ratio is also responsible for cell division, one says. So how is it possible, right? Whenever a cell grows, whenever a cell grows, whenever a cell grows, the time what happens over here, its volume increases. When its volume increases, but the surface area of the cell doesn't increase, it remains constant, so that cell has to divide, right? The time surface area, the time volume of a cell increases. Your volume of the cell increases, the surface area remains same. So that what happens? The cell divides. The cell divides. This is also one more cause for cell division. Surface volume ratio is also one cause, and other causes, mitogens. So all these are the causes of cell division. And one more thing I had explained in the last video. So in this video also we'll see the same factor, which is also one of the causes of cell division. What is that? So there are some enzymes like kinase. That is, already I have mentioned CDKs, cyclin dependent kinase proteins, right? So these are also driving the what? Cell division or cell cycle. These drive the cell cycle also. Many of the scientists, the one who worked on the division of the yeast cell, right, and some other cells, right, that moment they came to know there are some proteins. Cyclin is one type of a protein, so which is responsible for the cell division when it combines with cyclin dependent protein kinase itself, right? So that is one more factor, so which is also the major cause for the cell division itself. So I shall explain that cycle, how, so these are responsible for the cell division. Yes. So there we can see this diagram. In this one is CDK, another one is cyclin. So these two together, what they are responsible for the mitosis. These are factors responsible for mitosis. See here, what happens here is cyclin degenerates. It goes to the combination of, it leads to the combination of cyclin with CDK. Simply what remained here, CDK remained over here, and this CDK combines with another protein called as S cyclin. S cyclin. This leads to the formation of the combination of S cyclin with CDK. With CDK. So once again here, the S cyclin that degenerates. S cyclin degenerates over here. It degenerates into several pieces. So once again, what remained over here? CDK remained over here, and it combines with, once again, it combines with one more cyclin, that is M cyclin. Combines with M cyclin so as to form MCDK here. Right? These two forming the MCDK. Right? So this is how cyclically the cell division occurs due to the stimulation of these factors itself. Right? So here, during these stages, there we come across two checkpoints. Two checkpoints we come across. So these checkpoints occur usually between G1 and S. Here, whenever the transition from G1 to S is taking place, there one checkpoint we come across over here. This checkpoint is the first checkpoint. This is the first checkpoint, right? So that helps in transition from G1 to S. And another checkpoint that occurs usually from G2 to M. G2 to M. This is the second checkpoint that we come across over. This transition from one phase to another phase is controlled by these CDKs or cyclin, where it combines with CDK itself, cyclin dependent protein kinase itself. When they combine themselves, they form a factor, so that helps in transition from one phase to another phase. So there are two checkpoints are present in the cell cycle. One is in between the G1 and S, and the second one is G2 as well as M phase. Yes. After causes of cell division, let us go to the next factor, that is types of cell division. Types of cell division. What are the types of cell division? There are three major types of cell division that we come across. So one is amitosis, and second one is mitosis, and third one is meiosis. Are the three types of cell division that we come across over here. But actually there are two major types. One is mitosis as well as meiosis, and amitosis is also one more type of cell division. So amitosis is also said to be as a direct cell division, and mitosis is called as indirect cell division. Right? So amitosis, let us start with, is also known as direct cell division. It is usually occurring in lower organisms. So it is usually occurring in the lower organisms, especially this was first discovered by discovered by one of the scientists, Raymond. Is the name of the scientist the one who discovered this amitosis? So in which what happens initially, if this is the cell which contains the nucleus, this is a cytoplasm, right? Nuclear membrane, this is the nucleus, this is cytoplasm. If the cell is like this, it undergoes division. How it undergoes division? Basically, the elongation of this nucleus that occurs over here initially. See here, if the cell is like this, there initially the elongation of the nucleus that take place, that leads to the formation of a dumbbell shaped nucleus. Nuclear elongation take place initially, and this leads to the formation of totally dumbbell shaped nucleus. This is dumbbell shaped nucleus, and this leads to the formation of two nuclei within it. Two nuclei within it. Ultimately this leads to formation of a constriction of the cell. It leads to the formation of two individual cells. Two individual cells. So these cells are called as daughter cells. These are daughter cells. These daughter cells are the identical copies. They are the identical copies of parent cell or mother cell. They are the identical copies of the mother cell. So this is how the nuclear division that occurs first, and later the cell division occurs. It is followed by the cell division. So this is how the amitosis usually occurs in lower organisms usually. Example, PPLO, right? You know PPLO, right? And even it occurs in a bacterial cell. In some of the eukaryotic cell, like some fungi, right? In some fungi like yeast, so yeast budding is taking place over here, is also by amitotic cell division it is occurring. And even in case of amoeba, that is multiple fission occur. Multiple fission in the sense that a single amoeba cell that divides into many number of amoebas here. So this is said to be as multiple fission, it is said to be as. And this multiple fission that occurs in amoeba, it is by amitosis itself. So these are best examples for amitosis. This is one type of cell division that is called as a direct cell division, which was first discovered by the scientist Raymond, right? So and second one is mitotic cell division. Come to the next type of cell division, that is mitotic cell division. It is called as indirect cell division. It is called as indirect cell division, and it is also called as equational cell division. And this mitotic cell division is called as equational cell division. Why? Because so here, if this is one cell which is having a diploid number of chromosomes, this cell divides into two equal sized daughter cells which contain the same number of chromosomes in it. So that's why here from this cell the same number of chromosomes are shared between two daughter cells. So it shows that equal number of chromosomes are present both in parent cell as well as in daughter cell, right? Hence it is said to be as an equational cell division. It is called as equational cell division. And such kind of mitotic cell division was first studied by Fleming. Fleming is the name of the scientist, the one who studied this one in one of the animal cells, that is salamander. In salamander cell, it was studied for the first time. And after himself, in case of plant cell, so another scientist, Strasburger, is another scientist, the one who studied the same thing in plant cell. In plant cell, such kind of mitotic division, so that differs from amitosis. How it differs from amitosis, that matters over here. See here, in case of amitosis, there is no spindle formation. There is no spindle formation that takes place over here, and there are no distinct chromosomes. That are formed during cell division. But in case of mitosis, what happens here is, they are the spindle formation. The spindle formation take place. Along with that one, there the chromosomes are formed as a result of the condensation of the chromatin material present in the interphase stage of the cell, right? So these chromosomes are formed over here. And these chromosomes, which contain the DNA or the genes, the genes, so they duplicate during interphase. So they duplicate during interphase, right? So this is how amitosis as well as mitosis, they are different in. And so in case of mitotic cell division, there we come across two phases. In mitosis, there we come across two major phases. One is called as interphase, right? And second one is called as M phase, or it is also called as division phase, where the cell is supposed to divide itself. Interphase, so we have studied this phase in detailed manner. Basically, it was said to be as the resting period, but later we came to know it is not at all the resting period of the cell. It is actually the preparatory phase of the cell, during which what happens here? During which what are the proteins required? What is the DNA required? All have been multiplied along with the cell organelles required over here. So in interphase, all these are the aspects that we have studied previously. In M phase, there we come across Once again, two phases there. So one is karyokinesis, means nuclear division that we come across, and it is followed by cytokinesis, that is cytoplasmic division, right? So these are the two stages that we come across in M phase of the division phase of mitosis, right? One is karyokinesis. Karyon means nucleus, so kinesis means breakdown, the breakdown of the nucleus initially that takes place, and it is followed by the cytokinesis. Cyto means cell or cytoplasmic content, and kinesis means breakdown, equal. So amount of cytoplasm is distributed between the daughter cells that occurs over here. And this cytokinesis is somewhat different in case of animal cell and in case of the plant cell. What is the major difference between plant and animal cell during cytokinesis? There will study in detailed way in the next video. Thank you.